Hi there, and welcome to the Principles of Dynamic Causal Modelling. My name is Arthur Mitchell. I'm a research fellow at the Department of Imaging Neuroscience at UCL Queen's Square, and also a visiting postdoc at the Francis Crick Institute. This lecture is for the SPM for MEG/EEG course run by UCL, and I'm here to talk to you about dynamic causal modelling. To start, a quick disclaimer. I don't actually work with MEG or EEG data, but I do work with DCM. So this talk is going to be about how you can use dynamic causal modelling, how it works, and its more general applications across neuroscience. There are lectures specifically on DCM for MEG and EEG, but I'm just here to talk to you about how DCM works. So, a bit of history about DCM. It was really developed through the 90s by Professor Carl Friston, and that culminated in this 2003 paper by Friston, Harrison, and Penny. And if you want to learn a bit more, this is a good place to start. It's a very good paper, and in that, DCM was developed. This is a picture of Carl here, I think around the time this was written. So it was first developed for modelling fMRI data, and then later used for EEG and MEG, and you should also go and read this 2008 paper by Keeble et al. For a summary of how it was adapted in 2008. Even later on, it was used beyond neuroscience, which is quite a recent development, and at the end I'll just touch on one of the use cases, but there's several others in development which might be interesting for people to go see if you'd like to see a wider range of how this modelling framework can be used. So, what is DCM? Well, my favourite description of it was by Professor Peter Zeidman, which is a generic modelling framework for separating causes from consequences. So it's a form of generative modelling. It's a way to answer questions in a formal and evidence-based way. If you want a very practical definition, it's a tool or a framework housed within the SPM toolbox, which is primarily written in MATLAB, although we have recently released a Python version of it, and I think there's a couple of versions in other programming language. But most of the development is done in MATLAB, and certainly for EEG or neuroimaging use cases, it's primarily using MATLAB. But for me, I really view it as the key thing for DCM is it's a framework for testing hypotheses. So if we have a problem and we have a few hypotheses of what the best explanation for the data we've collected in, DCM is very, very useful for testing those hypotheses. So this is a pretty quick introduction to what DCM is, but essentially, if we have a hypothesis and we have a model of how something works or an idea of how something works, we can use DCM as a modeling technique to test how well the data we have explains this. And for a fuller explanation and description of what you can use DCM for and what it is, feel free to check out this page in the SPM docs for a much more in-depth review rather than the 2 minutes I'm spending talking on this slide. I think it's also useful to say what DCM is not, and it is not an exploratory approach. So if we are just giving it data and trying to say, find something and we're trying to find new things without a hypothesis or without some knowledge we're bringing to the table, DCM is not going to be very useful for you. It is also not a classifier. So if you think of sort of a lot of deep learning or machine learning applications where you sort of have two groups and you're trying to get the model to distinguish between them, then it is not particularly good for that either. You should not use it for that. It is also not a data-driven approach. Again, you're going in with a hypothesis. So you're not giving it the data and then trying to work backwards from there. You're going in with a hypothesis and you're seeing how well the data supports that hypothesis. And it is not a general purpose tool. You want to use DCM for specific cases and for specific hypotheses or problems you are trying to find evidence for. So I hope that helps in the distinction of what you typically use DCM for. So back to its original use cases. So we often use it for estimating connectivity in neuroimaging, and I'll just walk you through a few different types of connectivity. So structural connectivity is the axonal connections in the brain telling us which regions are connected. So it's describing physical connections in the brain. And this is a completely made up model, a diagram that I've come up with. So if we take this brain slice and I've picked 4 random regions of the brain and said that they're all fully connected, and this is our structural connectivity. This is our idea of how the brain is connected and what parts are talking to what other parts. And it's just describing physical connections. We also have functional connectivity, which is statistical dependencies or correlation in the brain over time. So this tells us which brain regions work together. And an example of this is if we take this original model, and perhaps there's a stimulus between these two. So if we have 3 time points and there's a stimulus between time point 1 and time point 2, we can look at which brain regions of the brain work together and how they correspond, and look at things like this. Now you might think, do we need DCM to do these two things? And the answer is no. But we do need it for this third type of connectivity we can look at, which is called effective connectivity. So this is directed causal inferences between brain regions and answers questions such as how does a change in one region alter another? So for the previous 2 types of connectivity, we are estimating or we are looking at data, but we cannot interrogate specific cases and specific hypotheses and do specific tests. So we can, but when we look at effective connectivity, we can uncover causes and effects in our model and data. So perhaps if we're interested in this region here, we can use DCM to modulate it and look at how it affects other regions of the brain over time. So DCM is a way to solve something called the inverse problem, which some of you may have heard of. So we're trying to find the causes of some data, and this is comprised of a forward and reverse or inverse problem. So if we start with our brain and we have some method to measure the brain, which is represented by this arrow here, so it could be MEG, EEG, fMRI, and that produces some data that might look like this. So this is some MEG data, and this is going the forward model. So this is going from cause to consequence. So the brain is causing something, and this data is the consequence of it. But the more difficult and possibly more interesting question is, what is the inverse model? So the cause is often unknown, so we only have the consequence really and some idea of what the cause is. So the inverse model is going from consequence to cause. We have this data and we want to use it to figure out what is going on in the brain and what is driving this data. So how do we do that? We first must hypothesize. So we must start with some theory about brain function. So perhaps again, this is our model. This is our theory of how these brain regions are connected. And I'm just going to pause for a second and talk about DCM and how it is not all about comparing models. While a lot of DCM is about comparing different models or perhaps changing them a bit or looking at what covariates are important in a particular hypothesis, you can just use it for a single theory or model and just use it to estimate the parameters or the connectivity of that single model. So if you're really confident about your model, you may wish just to have a single model, find their completely inverse problem, and get your connectivity, and perhaps do some simulations or generative modeling based upon that. However, a lot of the time, we have multiple hypotheses, and we're looking for the best explanation of the data we've collected. So if I have this, and this is model 1, and then I have another theory about how whatever function I'm investigating works, I can call it model 2, and perhaps it looks like this. So it's not quite as connected, and we're assuming that this sort of, you have to go through each brain region like this. And this is defined as a mathematical model of a system of y, which is our observable data, x is our latent states, theta are our model parameters, lambda is our uncertainty, and m is the model itself. And as y is actually the only thing we need to collect and put into the model, this can actually be defined as y given these other model parameters. So once we have our definition of our mathematical model, which is our theory contextualized in a mathematical process, we can then ask the question, which model explains the data the best? So where do we go from here? Well, we do need some data to conduct this modeling, so we must design an experiment. So your design will vary depending on the hypothesis and what you're trying to prove, and you should really consider using a factorial design when you do this. So a factorial design, for example, if you had 40 participants and you had 2 factors you're looking at and each factor had 2 conditions, you might divvy them up like this. So if we take 10 participants, look at factor 1, condition 1, factor 2, condition 2, and then for the next 10, we use factor 1, condition 2, and factor 2, condition 2. This is a good way to explore all the factors we're looking at in our experiment and our hypothesis and get a good amount of data and also look if there's any effects between the two. So this was really developed for crop science, in fact, in Rothamsted, which is still quite a big agricultural research centre up in Norfolk. And it's very effective, and it works later on with our second level group analysis, which I'll come to later. So we've designed our experiment perfectly for our use case, and we've conducted our experiment, and now we have some data. So we've collected it, and now we need to pre-process it. So this is just taken from the SPM website. Again, we might have some time series imaging. In this case, they're showing an fMRI example, but we need to align it, normalize it, and perhaps soothe it. Again, this will depend on your imaging technique and your experiment, and you should consult for your specific use. But we need to do some pre-processing. So everyone's data is the same, and we don't have any sort of differences between machines or anything. So yeah, we need to normalize and align. And again, this is going to differ between your region of interest and your imaging technique. We also need to give the model some priors. So these are estimates for the parameters of Latin states that we're trying to measure and predict. So with these priors, these are reasonable estimates you have for each parameter. We need to do this so the model has no concept when you start it of what a reasonable estimate is. It could, if you gave it perhaps infinite priors, it could come up with some absolutely insane, physiologically impossible results. So we need to give it priors so it knows where to look and what's reasonable. And these are expressed as a Gaussian distribution with a mean of variance, and that's because we really want it. We want the model output to be somewhere in the mean. We don't want to be pushing it to the extremes. We have a sort of a theory, and we're looking to see how close to that we can get. And if you're very confident in a parameter, as you give it with a mean of variance, you can specify a low variance if you're very confident. But if you're not as confident, you may want to specify a high variance for your parameters. So there's a wider range of values that could be. An example of this is if we have this model, we have parameter A, parameter B, and we're very confident in A, we give it quite a low variance. But if we're not very confident in B, give it quite a high variance. There's a wider range of values it can be. And ideally, we want fairly low variances because we have an idea of what our parameters should be. And if we have this sort of very high variance, there's a wide range of what it could be. And that becomes more difficult for the model to solve, and you get a much wider range of outputs. This is also known as a strong prior and a weak prior in our model. So if we have our model and we've set our priors, we do need a way to evaluate it. And this is called the model evidence. So it is defined by how likely the data could have been generated by the model, given its set of parameters and data. So this is defined like this, where y is our data and theta is our parameters. And if you're familiar with Bayesian statistics, you may have come across this before. Unfortunately, this will only really work for small models and is typically not applicable for DCM, which is larger and more complex. So because of this, we have to approximate the model evidence. And how do we approximate the model evidence? Well, we use something called free energy when the model is too complex to just perform that calculation. So what is free energy? It is, again, how likely I'd see my data given the model, but it also adds a complexity term to it. So to quantify our model's explanation of the data, we use free energy of the model, which is also known as the evidence-based lower bound, or ELBO in machine learning, and that is defined like this. So the free energy is approximately equal to our model evidence, which is defined as our accuracy given our model minus our complexity given our model. Now, you might be thinking that perhaps if you're like me and you came first learned deep learning, why is complexity such a bad thing? And that's because we're really looking for our models to be explainable. if we have millions and millions of parameters, again, like in deep learning, those parameters end up not meaning anything. They're just something in the model. But in this case, we really want to understand our model. And we're trying to, again, I keep saying it, but we are trying to look at our hypothesis and prove or disprove it. And this is why this complexity term is important, because we want to limit the complexity of the model. We don't want these really complex models that we can't explain. We want a simpler, explainable model that explains our data. So if this is also, you may have also know of AIC or BIC. This is similar to that, but the key difference to those, while the accuracy metric is the same, and I'm not going to talk too much about it, the real sort of difference in free energy is the complexity term. And the complexity term is defined like this. We have our callback Lieber divergence or KL divergence, and we're looking at the prior distribution of the parameters, which is P here, and how our approximate posterior results, Q here, have changed from those priors in free energy. So if you remember, we have them both. We have our priors as a Gaussian distribution with the mean and variance. We have the same for our posterior. So when our model is inverted and we have estimated our parameters given their data, this is what it will output. And this is how we calculate our free energy. So an example of how KL divergence will change with free energy could be like this. So if this red circle is our prior and this green circle our posterior, and this is the output of our model, this will have a low KL score. Our prior and our posterior will not move very much. And I must give credit to a Will Penny presentation I've borrowed this from. But if perhaps our posterior becomes like this will give a high KL score. So we have a large move from our prior, and we define this as high complexity in the model. And this is, of course, for every parameter in the model. So if we have a lot of parameters and a lot of covariates, this will probably increase the complexity. As a result of this, we can make a likelihood ratio. So if we have model 1 and model 2, and we have our results and our free energy, this is our model evidence, and if we take the log of this, we get our free energy, and we take the logs and divide, it becomes a negative. So our Bayes factor is computed by the free energy score of model 1 minus the free energy score of model 2. And if you're familiar with Bayes factors, you can look them up and see if it's a strong Bayes factor. So for example, a Bayes factor of 10 would mean the model 1 is 10 times more likely to explain the data than model 2 is. And you can look them up. It's incredibly well established in literature. I believe just even looking at the Wikipedia page gives you quite a good summary of it. So we have a way to score and evaluate our models based on our data. But another example of complexity. In this case, we assume complexity is the number of components in a polynomial model rather than the changing of our parameters. And if I've just plotted something quite simple here, in that the green dotted line is our ground truth, and I've just plotted some points along there with a bit of added noise on each of them. I've then plotted a very simple linear fit, which is absolutely terrible at fitting the data and doesn't capture it very well. I've fitted a 12 component polynomial fit and a 20 component in red. And we can see with the points we have, the red line fits them incredibly well. Whereas if you go by the individual points, the purple line does not fit them as well. However, the red line is considerably more complex than the purple line and will be penalized for it. So when we calculate the free energy for the purple line and the red line, the purple line will have a much better score because of its lower complexity while still maintaining good accuracy. And this is really the trade-off we're trying to establish, is making our model simpler, which would presumably be less accurate, although that's not always the case. Would this be better than having a more complicated yet more accurate model? So we have our model, we have our priors, and we have a way to assess model fit. So how do we invert the model? How do we get these parameters? Well, we can now invert it to link consequence to cause. So if we go back to this model I took at the start, we do this part. We invert the model with our data and our hypotheses, and we see how well this fits our original data that we have. So how do we do that? We use something called variational Laplace, and I'm really not going to go into detail on this because it will take me a bit of time to do, and I think it might be covered a bit later on in the course, but essentially it's a model for an algorithm for model inversion, and it approximates the posterior distributions over the model parameters, again, giving you Gaussian distributions of those. So estimating the parameters of the equation using the model evidence. And this is handled by SPM DCM estimate. There are some other SPM functions, I think specifically for EEG models, but you would have to check the documentation for your specific use case and your specific data. But essentially, I think for an overview, that's all you need to know. If you were interested in going into more depth about how this works, I can recommend this GitHub page by Alex Lepoivre, called Variational Laplace for Dummies. You can find the URL here, or I think if you just type it to Google, it should come up. So if you want to spend a bit of time, get a better understanding of how this works, I think it's quite valuable to do. In addition, I believe Pranay, who's lecturing later today, has contributed to this book. So after variational Laplace, we also need a way to encode our data. And to do that, we use a general linear model. So the SPM docs say this is to identify where the brain experimental effects occurred. And yeah, we encode our experimental modulation using a general linear model. This can contain where certain stimuli happen and other data about participants, such as age and other covariates. We can also encode our system inputs into the GLM, such as visual inputs or button pressing. That's all done here. So your GLM will have the following matrices. A is your connectivity matrix, B is your condition-specific matrix, C are your inputs, and U your experimental inputs. So C, by inputs, I mean if you're doing a vision processing task, it would be where your visual information inputs the model. So the region of the brain that's in, or similarly for a hearing task. And this is where we encode it. And I'll have an example later on in this talk. So this brings us to our first level analysis. We have data from our first level, an individual, and we have our two hypotheses for the data. And we can take this individual's data and we invert the model, get the free energy score for our two hypotheses, and we can compare it. So if our free energy score for model 1 is -3 and free energy score for model 2 is -1, because you can have negative free energies, of course, which is better? Well, -1 is higher than -3, so model 2 is better. It's a better explanation of our data on this individual. In addition to that, we can also then evaluate their model parameters. So if we have two parameters, and I'm simplifying a bit here because you will almost always have more than two parameters, but if we have parameter 1 here on this axis, which is about 3, and you can see it's got quite a tight variance. It doesn't vary much. So we're quite confident in this parameter. And also note I've plotted the same axes for these two parameters to highlight the high and low variance. Parameter 2, however, which is about 1, has a huge variance, so we're less certain of this parameter. There's a wider range it could be, which would have a much higher complexity. An example of this in literature would be this paper by Kumar et al., where they were looking at the hierarchical processing of auditory objects in humans is the name of it, but they were looking at how they had some theories about how we process auditory information in the brain using fMRI. And they came up with 16 models with their input here and tested them all using DCM. They did score with AIC and BIC, but mostly this is, I think, always pretty much how it uses free energy. And we can see at this first level example for individuals, each of these different subjects, that model 1 seems to explain the data the best in most cases. So you could perhaps use this as an argument for this is the best model you have of auditory processing in this case, or whatever kind of function you're trying to estimate or hypothesize about. I'd highly recommend looking at this paper, even if you sort of just skim the abstract results and the figures. because I think it's a very good use case of how DCM could be used to estimate how the brain works. So if we've done our individuals, which is our first level, we can then move into our second level DCM analysis. So in this case, we compare group level data. And we can take the model further, try new combinations to assess model fit, and generally we're going to get rid of outliers. So you might just have one result where someone's brain is just a little bit different compared to the data. We saw that in the last slide with that example where for most cases that model explained processing the best, but for some people a different model was a better explanation of the data. So to analyze at the group level, we use parametric empirical Bayes. So we use this equation to do that. And what does this equation mean? Well, if we take one parameter for each subject, so we do this for all of the parameters, and we have our design matrix, which will be of size subjects by covariates. So if we have 3 subjects and 5 covariates, this will be a 3 by 5 matrix. And then we are trying to get this group level parameter. So we are taking this parameter from each subject and getting it into this parameter for the entire group. And then we will also work out the error doing this. So there will be some error on subjects from measurements, from variability, whatever it is. This we will calculate the error, which is important because it will also tell us how much of our data can be explained by variability. So a short coded example is if we have U as our experimental data, so we have some stimulus, some attention condition, and we have a 30 by 2 matrix. So we have two regions, and that's our actual data here from those regions. We will have some intrinsic connectivity, so this is our model, connectivity between 2 regions, and in this case we're just having region 1 going to region 2 and nothing in the reverse. We want to look at our attention strengths, so we're modeling the attention on region 2, and we have our input going into region 1. So in this model, what we're saying is in region 1, we have it going into here, into V1, and then it's connected to V2 in one direction, and we're modulating the attention of V2 to see how that alters. So this is the parameter we're trying to estimate and how that changes with this model. And DCM uses a struct structure when you're programming, if you might be familiar with the MATLAB. So you do DCM.A, .B, and .C, and .U, which is your data. And then you can fit it using this function called SPM DCM estimate. And this will also handle your GLM for you. So there are a few other DCM functions within SPM, I think specifically for EEG and MEG, but this will vary depending on your use case and what you're using it for. But generically, you can use this function. So at the group level, we have our model, and we fitted it to all our participants in our experiment, and we have the group free energy and our group model parameters. So we're explaining over everyone what the best fit is generally, rather than the individual. Now, while we've compared these models and we have our covariates, we may want to see every covariate is going to add complexity. And we might want to see if the reduction in complexity and presumable reduction in model accuracy, although this might not be the case, if we can explain the model better or explain brain function better. So to do Bayesian model comparison, we will remove a covariate and recalculate the free energy score. So does the reduction in complexity and possible reduction in model accuracy improve the free energy? So if it's a parameter that's not really, or covariate, sorry, it's not really doing anything for the model, and it's just adding complexity, this will increase our free energy score. So what does this look like? So we have 5 subjects of 4 covariates. We will exclude 1 covariate and have 5 subjects of 3 covariates, compute the free energy, and then we can do our free energy comparison and get our Bayes factor. And this is a very easy way to tell us what covariates are important and if we can reduce the complexity. We can also do Bayesian model reduction. So this is investigating the actual structural model we have, and we can do this if we want to estimate a better system. So if we go in with our full model, perhaps with more connections, we can go one by one and switch off each connection or multiple connections and then score the model. So we do this by fixing the parameter to 0 in the mean and standard deviation, so it has no effect on the model. We can fit the model without any of the connections. This might look like this. So if we're unsure of this connection here, we can turn it off, score the free energy, and reveal a better structure or a better explanation for the data. And we can do this with multiple connections. So in summary, we want to come up with a hypothesis, collect our data, select our features we're looking at with our collection, plan out our model structure, set our priors, invert the model, and then do our Bayesian model comparison and Bayesian model reduction. So towards the end now, so I just like to cover some examples from literature. And I really liked this study they did a few years ago when they were investigating PTSD. And not only did they use DCM to show how PTSD differed from healthy control, they also found differences between PTSD and a PTSD dissociative subtype. So if we look at the left and right hemisphere here in these diagrams, we can clearly see the effective connectivity is different, and the way the brain is working and talking to itself, talking to different regions, connecting, is clearly shown to be different in this example. So again, this is another nice example you can go through if you want an example of how DCM can be used to solve very complex problems in neuroimaging and provide comprehensive explanations of the data that's collected. We can also use it outside of neuroimaging now. This is a relatively recent development, but this model is used for COVID-19. And they did this back in the start of 2020 when the pandemic was just sort of kicking off. And they were using it to model how COVID would affect the population and how many people would be positive for COVID, how many people would die, and how full hospitals might be. So you can see this is from their first paper, and you can see the death rate, new cases, and ICU occupancy, and the cumulative deaths as well. And this is a model that has been refined, particularly by Cam Bowie and Cole Friston. And they've done a few iterations of it now, and I know recently they've been using it to investigate if mask wearing was how much of an effect that had on COVID-19 transmission. So I'm really showing you this to show that DCM, although it has clear roots in neuroimaging, it really can be used for a variety of uses, and it's a very active research area at the minute, looking for cases beyond neuroimaging. So finally, I'd like to talk to you about the SPM DEM tool. So if you go to your MATLAB and you just type in, if you've installed the SPM toolbox, you could type in DEM here. You may need to type in SPM fMRI or SPM EEG to initialize SPM, but you may already do it. It varies from person to person, it seems. But you can type in DEM. So after you type DEM, this box will pop up. And I know it looks very big and scary, and there's loads of options, which you probably can't see on this presentation. But these are lots of examples of how you can use SPM and DCM in research. So there's tons of different examples. There's examples of how variational Laplace works and some very specific use cases. There's also the COVID-19 model down here if you're interested in how that works. But it's good to have a look. And if you're struggling with anything, it may be worth, I know I certainly find it easier to look at a coded example. So it might be worth for you to go do the same. For me, it helps when I look through code and I can figure out how it works. There's two sections which will probably be most interesting for the people in this talk. And that is the variational Laplace and dynamic causal modeling section and the hierarchical empirical Bayes section. So this section has a lot of examples of how DCM can be used, primarily in fMRI, but there are some different types of DCM. There is a large DCM and how to do Bayesian reduction. So again, it might be beneficial for you to go look at how these examples work and have a good hands-on example of how you can use them. However, I suspect the main example and the most interesting one is simulated EEG analysis. So this example uses simulated data and it talks you through how you can solve a task. And you may want to replicate this in your own work. So this is a very good tool to have, and it's good to just see how you can code these things to work with your tools. Finally, if you want to look at the docs, there is a good tutorial here on the SPM docs. Excuse the very long URL, but if you go into the SPM GitHub page, you can find this EEG for DCM example, and it gives you a very nice, very in-depth summary of how you can use DCM in EEG settings. And of course, that is being covered later today. This is just the introductory lecture to DCM. So I hope I've helped your understanding of DCM. I'd like to say thank you all for listening. And if any of you have any questions, feel free to ask them, or you can indeed email me, raise an issue on GitHub. There are many ways to contact the SPM team if you have questions. So thank you all very much for listening. And if you have any questions, I'm more than happy to take them.